alaikum, dear students. After the extraction of DNA, now we are moving toward our second lecture. So once we are extracting the DNA, we want to check the purity of our DNA. Means we want to quantify the DNA. What is the quantity of our DNA that we have extracted? So for the quantification of DNA, we have to use two methods. One method is we quantify the DNA by using the spectrophotometer, and the second method is nanodrop method. In today's lecture, I will describe the spectrophotometer method of DNA quantification. So, what is spectrophotometer? Spectrophotometer basically it is an instrument that measures the absorption spectrum of DNA. So, basic principle on which spectrophotometer quantifies the DNA is on the basis of its absorption spectrum. So, spectrophotometer comprises two qubits, means two small tubes. Spectrophotometer comprises two qubits. One qubit acts as control, while the second qubit is used to test the sample. In simple words, one qubit has control sample, while the second qubit has the test sample. The test sample is that sample where we want to quantify the DNA. So in control, what control sample have? In control sample, with the help of micropipette, we only add 245 microliter of distilled water. While in case of test sample, what we do? We add 245 microliter of distilled water, and in addition to that, we add 5 microliter of isolated DNA. So, what is the working philosophy of spectrophotometer? In other words, how we quantify the DNA by using spectrophotometer, I will describe in this video tutorial. So, I'm just coming toward this video tutorial right now.
In this video, you will learn how to perform DNA quantification. There are many ways to quantify DNA.
So,
in this activity, the amount
this
of
is
DNA
spectrophotometer.
plasmid preparation will be estimated using a spectrophotometer.
So, this is spectrophotometer, okay, as I have described in my lecture. So, these are two qubits, okay. This is one qubit and this is second qubit. So, in one qubit, we used to put control sample, while in other qubit we used to place the test sample. So what we do, with the help of micropipette, we take 245 microliter of distilled water and put this double distilled water in each qubit. So now after what we do, we take 5 microliter of our isolated DNA. Now, what we do? On spectrophotometer, different options are given. Out of these options, we have to select, we have to press this button. Now, what type of nucleic acid, that is double stranded DNA. Now we press the button enter. So you can see the screen of spectrophotometer. After pressing the enter, you can see absorption spectrum. Now we further move. So now, first of all, what we did, we have taken our control sample, okay, and place it at the testing site. This is the testing site. Now after that, we have taken that sample that comprises DNA. Okay, now we place it in this cavity, in the testing chamber, testing cavity. Now we have pressed the button. Okay, now it is reading the absorption. It is measuring the absorption. So no, this is the reading. This is the concentration of our DNA that we have recorded here. So what we did in first step, we placed our control sample, because you have seen there were two cavities. This is cavity 1, this is cavity 2, and this is the place where, for example, control sample was placed here and the test sample was placed here. So this is the testing cavity, okay? Now, when we placed our control sample here, and we recorded the absorption, then afterward we placed our test sample within the spectrophotometer, and we have recorded the absorption spectrum. So definitely, because here this is only water and here what is present, DNA is present. So what is the difference between these two absorption? It means this is difference is just because of the presence of DNA. So at the end, when we have pressed the button, what we have observed the quantity of our DNA within that test. So this is the end of this lecture for the